Research and public relations need more financial and educational input from sources other than the seafood industry. All the groups worried about various forms of mercury, all for one, one for all, need to work together to ensure the health of pregnant women and young children, and there's no question, nutrition-wise, essential fatty acids and hazards, amalgam mercury, methyl mercury, and thimerosal need to be taken into account. So let me, in the few remaining minutes, discuss some work that's going on in our laboratory. The first is our search for a plasma protein biomarker for autism. I'm not going to give you background on autism. Jim Adams will do this very, very nicely this afternoon for you. All I can say is that in many ways, with many children, it can be a very terrible disorder. We have used a new technique in proteomics called differential in-gel electrophoresis. It makes me wish I were 20 years younger, in fact 30 years younger. This is really an amazing technique, and let me go through it once with you to explain and show you. First of all, you take a plasma sample, or you take blood, you spin down the plasma, and then you take a little bit of that plasma sample, and to it you add a dye. This dye that you add will fluoresce red when it combines with proteins. If it doesn't combine with proteins, it won't do anything. But if it combines with proteins, those proteins will fluoresce red. You can take a control, non-autistic, take his plasma, and you put in a dye that's going to fluoresce green when it links up to a protein, binds to a protein. You then we take 250 micrograms. We've even gone down to 10 micrograms of protein from each of these plasmas. You put them into a test tube, and you mix them. So you now have mixed sample 1 and 2. You then run what we call an isoelectric separation, which is according to pH in this direction, and then you do a electrophoresis, molecular weight size electrophoresis in this direction. If the spot is red or green or yellow, it's going to have a very significant meaning. If the spot is green, it means the autistic child has less of that protein than the non-autistic child. Or to put it another way, the autistic child has more. Both proteins from both of these individuals are in that spot. If the spot is red, it's going to mean the autistic child has more of that protein than the non-autistic child. If the spot is yellow, it means they're equal. We can do 1,500 spots. The software for this technique is incredible. The software is more sensitive than our own eyes. Now, these green spots indicate the autistic child at this spot. This autistic child had 81.5 times less of this protein in his plasma than the autistic child did. These other spots now, and this tells you how much some of these differences are. The software gives you a quantitative relative amount as to which one is more or less. Then you cut out these spots and you send them over to the mass spec facility, and they identify the proteins with you. Now, all these green spots that I showed you, there are other ones that we could point out, are essentially chains of a compound called haptoglobin. Haptoglobin, among other things, I'll tell you what it does in a moment, among other things, it's an acute phase protein that many hospitals do routine tests on. If you have a fever and show up in the hospital, they're certainly going to do, I think, a haptoglobin determination. But the point I'm making is we are not dealing with some very complicated assay that hospitals would cost thousands of dollars for them to put in. They already have a procedure for testing this protein. So what we did then was send the plasmas over to the University Hospital laboratory, and the plasma haptoglobin concentration of autistic children was found by this method to be less than non-autistic children. This is a control 73.6, autistic 29.5, determined by an nephelometry at the University Hospital. The P is 0.04, borderline. We need more people, obviously. The autistics were 3 to 10 years old. controls are 5, 10 years old. Criticism, autistic controls not age matched, and the N is small. But we are, actually we're downsizing the lab for a number of reasons, and we're probably going to stop doing this, but we're very excited by it. So, haptoglobin prevents iron loss. It has antioxidative activity and can modulate the immune system. And I go on more with this, but I won't. These are some of the other properties. We're also fortunate to have had someone give us a gift of $210,000 that we could buy the latest piece of equipment to determine metals in urine and metals in blood. This is an HPLC ICP mass spec. And so on all these children that we collected plasmas on, we also collected, once we decided to do this, urines on. And the beauty of this instrument is it can simultaneously determine 21 metals. Okay? It's compared to the old style, it's unbelievable. But what has always been consistent is that the mercury, urinary mercury of autistic children has always been less than the urinary mercury of controlled children. And the others sometimes, depending how many samples we have, one is more than the other, but the mercury has always been lower. Now, we don't quite know what to make of this. We'd like to think that this is proof of the mercury efflux problem in autistic children. We'd like to think, we have no hard evidence for this, there's a lot of circumstantial evidence. We'd like to think that the problem in autistic children is that they cannot get rid of mercury from their cells. Like Wilson's disease, you cannot get rid of copper from your cells. It's a copper efflux disease. But the other explanation of this could be that mothers of autistic children don't give them any fish to eat because there's mercury there. But mothers of non-autistic children still probably give them tuna fish. So we can't say with certainty what this means, and we need to do a lot more. What has to be done, and has not been done yet, is to take tissue, brain tissue that's in tissue banks of autistic children, and determine whether they are high in mercury or not. So very quickly, let me go through our brain mercury data. Again, this is because of Jim Adams. He's such a nice guy. He's going to give you a talk this afternoon. It's very hard for anyone to say no to Jim Adams. Jim had heard that penicillamine, we published the first paper on penicillamine and mercury way back in 1957, I think it was. And Jim wanted to know, will penicillamine decrease brain mercury? So therefore, it might be useful with autistic children. And so the first question was, will it decrease brain mercury? The protocol essentially give animals 10 milligrams methylmercury in their drinking water for 28 days. Drinking water for 7 days, we have increased this number in other experiments quite a bit. And then we give them deep penicillamine twice a day. We inject it, although you can give it orally. But please note, it's the D isomer. The DL isomer or the L isomer will cause a severe pyridoxal phosphate deficiency. This has been clearly shown by our lab in the literature and other labs also. So if anyone's going to fool around with this, which I don't recommend yet, they should not fool around with the L isomer or the DL racemic mixture. And so essentially what you see here is that in these experiments, deep penicillamine decreased mercury in brain and kidneys of mice exposed to methylmercury. And here you can see quite a difference, as you can see here, the control with the kidneys. What we have not done yet is to see, Dr. Berlin, who's in the audience, was one of the first to show that if you give British andalucite to people or to animals with mercury toxicity, that there's an initial increase in the brain mercury because of the redistribution. So we have yet to see whether when we redo these experiments and kill the animals at day 1, day 2, and day 3 of penicillamine therapy, whether the brain mercury has increased and then it will go down. We don't know yet. These are the two students involved in these experiments. The work in our laboratory has been supported in part by a variety of organizations. The Wallace Research Foundation in particular has put in over $1 million into our lab over the years, a number of other organizations. Let me summarize. The FDA amalgam panel was a success, I think. The important mercury forms, their interrelationships and mysteries must not be forgotten. And remember, all for one and one for all. Selenium, mercury, and seafood, the ratio is important. D-penicillamine decreases brain mercury. A conversation with Michael Ziff I'll take up in a moment. As I said, Michael and I used to talk. And if Michael were here today, I would suggest that the IAOMT consider these scientific goals. Develop a test to detect people who are hypersusceptible to mercury. I'm not talking about hypersensitivity, hypersusceptible toxicology. Determine whether there are any specific signs of micromercurialism. Determine single nucleotide polymorphisms, SNPs, whether single nucleotides in your genes can cause certain enzymes involved in mercury metabolism to change their activity. Fund young investigators to study amalgam mercury. The strength of American science has always been in its young investigators. When I was in Russia in 1977, I met assistant professors who were 55 to 60 years old. I met the director of the best institute in Russia, who was a Nobel laureate, 82 years old. Waiting for him to retire was a deputy director who was 78 years old, so he could come. So our strength in America has always been our young scientists, and we should try to support them as much as we can, especially in these very bad days. Use top-notch investigators to judge applications for IAOMT-sponsored research. I had two other items down there, and I couldn't help but feel that Michael would have said, Vas, keep your mouth shut while you're ahead of the game. But I thought I might say this, that what we need are competent dentists working with competent biochemists, toxicologists, physiologists. And one of the best examples I can give you of this was Murray Vimy working with Fritz Lorscheider. Now, both of these guys are difficult guys to work with. I'm sorry, Murray. People say I'm difficult to work with also. But the fact remains that those original experiments turned on a lot of people to look into the amalgam problem. And we need more groups or more couples like that of young people. Murray's old now. We need young people who can use their young thoughts before their scientific blinders are put on. You've had this disclosure. Thank you for your attention. Let me again, before you clap, let me just say that there are three men in my life who passed away who had quite an effect on me as far as their passing away. One was my father, who died 27 years ago. The second was Michael Ziff. When he died, I felt I didn't have the communication that I once had with someone that I had bonded with. And the third man was Henry Wallace of the Wallace Research Foundation, who financially supported more medical research on mercury than even our federal government did. Thank you for your attention.
Don't know Michael if you've joined in the past 3 years, and he is what Vaz said and what everybody else has said, that he was a very dedicated, loving, funny man. But there was also a Michael Ziff that your executive director Kim and I lived with day after day. He had his own, I wanted you to know that he was a human being. We have a tendency when someone dies to turn them into a saint, and he wasn't. He had his own filing system that only he could find. Instead of filing things under topic, he filed them under author. So that when one of you would call and ask for the research, we would ask, who's the author? Well, I don't know. That's why I'm calling you. And we couldn't find it. I don't know how many times a day he would ask Kim and I where we hid his glasses. He swore the cat stole his socks and that I was hiding his keys. In other words, he was just a normal person trying to make a dent in the mercury issue to better the world. And the bottom line is, he was my husband, and I miss him. And so, when Rich told me that you had chosen to remember him with the Michael F. Ziff Memorial Lecture Series, I was thrilled, and Dad and I were touched. And it couldn't have been better to choose Vaz as the first speaker of this series. And so, because of the honor, we would like to honor you. Number one, I'm honoring you because in 23 years, this is the first lecture I have ever sat in on. And also, the Academy would like to honor you with this plaque that reads, "The IAOMT presents to Vasken Aposhian, PhD, in Tucson, Arizona, March 16, 2007, the Michael Ziff Memorial Award for unbridled dedication and uncompromising integrity." And we present it to you today, guys.
Thank you. Thank you, Lily. Thank you. You're
Oh, I'm shaking like,
wonderful.
if you'd like to say something.